This pre-lab covers experiments 4, part 1, part 2, and part 3 in your lab manual. In part 1, we're going to ask, does the substrate concentration affect catalase activity? So let's look into this. So catalase is an enzyme. And enzymes, for the most part, are composed of proteins that have primary through at least tertiary structure and have a very specific shape. So here is the active site. The active site binds to a substrate. In the case of catalase, the substrate is hydrogen peroxide. The substrate binds to the active site and the enzyme will actually change shape. Remember, structure is vitally important for function in proteins as well as all levels of biology. And the substrate attaches to the enzyme and the enzyme will actually change shape, forming the enzyme substrate complex. Now what is happening is that substrate is having its bonds weakened, or perhaps two substrates are being brought together so that the activation energy required for a reaction is now lowered. So this takes place in a cyclic fashion. We can call it the catalytic cycle of enzymes, where there's one or more substrates, in this case two different substrates, that bind to the active site, forming the enzyme substrate complex. The enzyme changes shape in response to this. The substrates are held together by weak interactions, perhaps ionic or hydrogen bonds, something like that. The activation energy is lowered because the chemical bonds within the substrates are weakened by the presence of the enzyme. Substrates are converted to products and then discarded. But the enzyme, as we can see here, is available for new substrates again. So this is a cyclic reaction. The enzyme is not used up by the reaction. Just like when you build a house and you use a hammer to pound in nails, when your house is done, you still have the hammer. So the enzyme is like a tool. It is used over and over and over again to run these reactions. And virtually every chemical reaction in your body is catalyzed by one or more enzymes. Catalase, the enzyme that we're looking at this week, has a very complex structure. We can see it here, lots of alpha helices and perhaps a beta pleated sheet or two in there somewhere. Quite a complex structure, and here is the active site. The reaction that this enzyme catalyzes is two hydrogen peroxides will be catalyzed by this reaction to break down. So we're breaking H2O2 down. So a catabolic reaction, an exergonic reaction, and we're yielding two water molecules and molecular oxygen. And one catalase enzyme can break down millions of peroxide molecules every second. It's a very active enzyme and very, very important for life. This is a highly conserved enzyme found in virtually all life forms that encounter oxygen. It's conserved because if you change it much, it doesn't function very well, and the organism would then die from the reactivity of hydrogen peroxide. So peroxide is actually a byproduct of cellular respiration. So we produce it, and then we have to break it down, and we use catalase to do that in our cells. You'll do a demo of liver in lab to see the action of this enzyme and to feel the exergonic effects of this reaction. The basic setup this week will use a pressure probe, a gas pressure probe. You will have some yeast. Yeast will be our model organism. It's a great organism for the study because easily available, and it's a eukaryotic cell that encounters oxygen on a regular basis. Therefore, it has peroxisomes that contain catalase, and we will be reacting that yeast with hydrogen peroxide and measuring the gas pressure produced as that oxygen is liberated from the reaction. So you'll need to form a hypothesis. What will happen as catalase activity, or I should say, what will happen as hydrogen peroxide concentration increases to catalase activity. We're going to measure catalase activity using pressure, in this case millimeters of mercury per second. So it's actually a rate. We're going to be measuring a rate for the first time in class. So a rate is time, is a measure of a change over time, and in this case change in pressure over seconds. So we'll be doing that in lab for the next couple of weeks. You're going to need to wear gloves and goggles this week. Remember, we're producing gas under pressure, and occasionally test tubes will crack or burst, and we want you to be safe. You're going to need to label things very well this week and not mix up pipettes. The pipettes that are used for yeast have to be different than the pipettes you use for the peroxide. And one thing I want to point out, we're pipetting yeast. Yeast contain catalase. Yeast is not catalase. Yeast is an organism that contains the enzyme catalase. Many times students mix that up and think somehow that yeast is catalase. And yeast is an organism just like you, only in this case it's a single-celled organism, but it has organelles just like you. And in the peroxisomes we have catalase, which will break down hydrogen peroxide into oxygen gas and water. The second week of experiments will involve changing the temperatures around this reaction and seeing how temperature affects the catalase activity. So how does increasing or decreasing temperature affect the rate of enzymatic activity? So remember, when making predictions on what will happen, that chemical reactions take place in an aqueous environment because molecules collide, and changing the temperature will affect the rate at which molecules collide with one another. And when you're determining your control for your experiment, remember, we're testing yeast. It is a eukaryotic organism, so figure out what an average temperature for yeast in the wild would be, and that would be a good place to start when thinking about a control. And then you can test temperatures above that and temperatures below that to see how activity will change. And in this experiment, you're going to heat up and cool both your peroxide and your yeast solution. You will heat and cool both of them to the temperatures that you'll be testing. The third set of experiments that we'll do is examine denaturation of catalase in detail. And you'll answer the question, is there a critical pre-incubation temperature where the loss of yeast catalase activity occurs? And if so, does the activity happen instantly or gradually as temperature changes? So is that protein breaking as soon as you cross a certain temperature threshold? Or does that enzyme slowly bend out of shape as hydrogen bonds and other bonds break as energy is added until the enzyme finally fails? So you will be testing temperatures of your choosing, trying to answer this question, and you'll most likely be writing a lab report depending on the plans of your lecturer and lab instructors. All right, that's it. See you in lab.